So what the nervous system? The nervous system is the system within our body that helps communicate with other and coordinate with other organs. Not only does the nervous system does this, but the endocrine system also communicates with other organs as well. But that's its main function. It consists of neurons, and neurons are the cells that actually provide that communication. They have electrical signaling passing along the cell itself, so it will actually pass along the cell using electrical signaling. And then from cell to cell, it will pass through as a chemical signal. So if it's passing the signal, it will be coming from this end, and then passing the chemical signal to another cell over here. So that's how they communicate from one cell to another. Glial cells are the other types of cells that are found within the nervous system, and they are supporting cells. So what they do, there's different types of glial cells. You don't have to know all of them, but you do need to know that what they are. Glial cells are supporting cells for the neurons, and they have different functions. So some of the functions: protection, insulation, and reinforcement. Yes, exactly. So here is oligodendrocytes, only found in the central nervous system. There is something very similar to its function in the peripheral nervous system called Schwann cells. They provide the myelination around the axons of neurons. So this is that insulation that we were talking about when we were talking about fast and slow pain fibers that we talked about in lab a couple weeks ago. So that's the insulation. So do you guys remember those that are insulated by this myelin sheet? Are they faster or slower in response? Yes, faster response. Okay, the nervous system are divided into the central nervous system, which consists of the brain, and also the spinal cord. Spinal cord is within our vertebral column. And the rest of the nervous system is called the peripheral nervous system. So the peripheral nervous system contains all these nerves that come off of the spinal cord, and some of them come off of the brain as well. And this is the rest of it. And the nerves themselves are bundles of neurons. So we talked about neurons in the last slide, right? It's just a single neuron cell, right? Nerve cell. So there's bundles of these that form the nerves themselves. There are different types of neurons I want you guys to know. One is the sensory neuron. Sensory neuron is part of the peripheral nervous system. So it carries information from the receptor. So in this case, do you guys remember what are the receptors within the eye? The photoreceptors, which they have two different shapes. Yeah, rods and cones, remember? So those are the sensory receptors in the eye. We'll get to there. If you don't remember it, that's fine. And then they will send signals from that receptor to the central nervous system through the sensory neuron. And once we get to the central nervous system, it either might be the brain or the spinal cord. In this case, it will be to the brain. So in the brain, it's going to go through these interneurons. The interneurons, like the name implies, is between neurons, right? So there's information being sent by the sensory neuron, and then there will be information that will be sent out of the central nervous system through the motor neurons, which we'll get into the next point here. So the interneurons, once again, is within the central nervous system. And what are the two organs that are found in the central nervous system again? Yes, the brain and spinal cord. Very good. And this is where they interpret that sensory information and then see whether or not they want to make some sort of response to that sensory information. Once they want to provide a response, then they're going to send it through this motor neuron. So the motor neuron, is in between. Going back, we have the sensory neurons sending information to our central nervous system, the interneurons that connect the sensory neuron to the motor neuron here. And the motor neuron sends information from the central nervous system to our effectors. And our effectors are these organs that actually performs that particular response. In some of our reflexes, we don't have this interneuron, kind of like that knee-jerk reflex that we saw in lab. That actually doesn't have an interneuron, so it's a monosynaptic reflex. Mono meaning one, synapse, which we'll talk about next, later, is just this connection between, or this gap between these two neurons. So it's a lot. Would you think quicker or slower if it was just a direct connection from sensory to motor without any interneurons? It would be faster, yeah, quicker. There are different parts of the neuron itself too. First of all, we have the dendrites, as you can see all over here. All these are dendrites all over here. And these dendrites, they receive signals from the either other neurons or receptors. And then they're going to send signals from the dendrites to the cell body here. And the cell body contains the nucleus and most of the organelles within the neuron itself. And then from the cell body, they're going to start sending information down the axon here. And is this axon myelinated or not? Can you guys tell? Let me take off the... Is it? How did you know it was myelinated? Yeah, you have the myelin sheath around it. Each individual white capsule that you see here insulates the axon to provide faster signaling. And the axon will then come over here, sending the electrical signals down its axon to the synaptic terminals here. And the synaptic terminals will contain these vesicles. Remember, vesicles are those membranes around these neurotransmitters. So inside of here are neurotransmitters here that the vesicles are encapsulating it. And then they're going to end up being pushed out, expelling the neurotransmitters into the synaptic cleft here. And what is that process called when the vesicles fuse with that plasma membrane and then expelling its contents? It was a type of active transport from our last lecture. Starts with the E. Energy? No, not energy. It uses energy, though. It uses ATP to do that process. What type of... It's something cytosis. Say again? Endo is taking in. Yeah, exocytosis. Very good. So it's through this process, through the synaptic terminal, synaptic bulb, whatever, they have different names for it, that they expel their contents of their neurotransmitters into the synaptic cleft here. So that's how they communicate with other cells. So remember, what kind of communication is it when they're communicate within the neuron itself? No, I mean, is it electrical or electrical process, or is it a chemical process? Electrical within the neuron itself. If it's from the neuron to another cell here, what is this? This is the chemical process, right, of communication. So the neurotransmitters is a chemical way of communicating. Okay, first we want to establish some characteristics of a neuron before understanding how they send that signal to another cell. So we have to first establish the resting membrane potential. The resting membrane potential is this potential energy that the cell has. So within the cell, the membrane here is the plasma membrane, right? So this is clearly not a neuron, but this is just a typical cell that we have in our bodies. And typically speaking, they tend to have a slightly negative charge on the inside of the plasma membrane. So it's slightly negative on the inside, slightly positive on the outside. Okay, so that causes this potential energy so that we can end up converting that into kinetic energy. So this negative energy is inside due to several things. Do you guys remember the charge of DNA? Is it positively charged or negatively charged? Negatively charged due to what? Do you guys remember? Not because of the sugar. Okay, it's not because of the deoxyribose or ribose. What parts of a nucleic acid do we know? We know of nucleotide, right? We have the ribose sugar, right? We have the nucleic base, so some base over here. And then, what else does it have? You guys remember this other thing here? Yeah, phosphate, right? And phosphate, yeah, the phosphate is negatively charged. So this is negatively charged. If it's negatively charged, it's going to cause the cell within the plasma membrane to be slightly negatively charged, along with other proteins and stuff that make it slightly negatively charged inside the plasma membrane. So that's why it's kind of slightly negative inside. Alright. Also, due to the sodium-potassium pumps, do you guys remember how many sodiums are pumped out and how many potassium are pumped into the cell? No. The sodium pump, 3 sodium out, while 2 potassiums in. So overall, we're going to have what? A more positive or more negative inside? If we're pumping 3 sodium out and 2 potassium in, right? We're losing a positive charge, right? If we're losing a positive charge, we're going to make it a little negative inside the cell. Does that make sense? Yeah. Okay, so now that we established those things that make it a slightly negative inside, the concentration of these ions also matter too. What is this? What is K plus here? Potassium? Yes, remember? No? It was during that chemistry lesson that we learned. Potassium is more concentrated inside the cell compared to the outside of the cell. So there's a lot more inside compared to outside. Concentration-wise, there's a lot more. What is this? What's Na plus? Besides Brian? Sodium, very good. So sodium tends to be a lot more inside compared to outside. Okay. Sorry, did I get it wrong? Yes, I did. It's outside is more than inside. Extracellularly, there's a lot more. All the other ions that you will learn, they're more concentrated on the outside compared to the inside. So potassium is the only ion that you will need to know that is more concentrated inside than the outside. What is Ca2 plus? Calcium, yes. Calcium is more concentrated where? Outside compared to the inside, yes. And finally we have Cl minus. What's Cl minus? Yeah, chloride. And so that's also more on the outside compared to the inside. Okay, I want to establish this because these are important when we talk about how a neuron functions. Okay, so we have a couple channels, and these channels, what kind of biomolecule are these channels made of? You guys remember? What kind of biomolecules are channels made out of? Proteins, very good. We have phospholipid plasma membrane, right? And we have these proteins embedded on the plasma membranes themselves. So there's a voltage-gated sodium channel here. And these sodium voltage-gated channels, they are closed at resting membrane potential. It's not until we reach threshold, we'll talk about threshold later, that it will end up opening. Okay, so here is my little artistic representation of a sodium-gated channel, voltage-gated channel. So right now it's closed, okay? And it opens up once it reaches threshold. Notice how it got a little bit thinner, so now the sodium can actually fit through. These are very fast channels, so they actually, once it hits threshold, it opens really quickly. Once at certain conditions are met, they close up really quickly as well. They also have an inactivation gate as shown over here. And it just closed up here. But this inactivation gate will block the sodium channel. If it blocks the sodium channel, then will sodium be able to go through it? No. Yeah. And okay, so going back a little bit, when it opens and hits threshold, which way will sodium pass through? Will it be going out or into the cell? Where concentration-wise, which side is greater? It's greater outside compared to inside, right? So in which direction will sodium go if this channel opens? Everything always goes from high concentration to low concentration, right? Yes? Because if it's passive, right? It's diffusion, right? So if it opens, more concentration on the outside goes into the cell where there's less concentration. Okay. Potassium channels. There's a couple potassium channels. The one that I want you to know is the voltage-gated channels. And the reason why these are voltage-gated is because once they hit a certain voltage, then it's going to open up or close kind of thing. So like I said earlier, the sodium voltage-gated channels, they open when they reach threshold at negative 55 millivolts. You don't have to memorize the numbers, but I just want you to understand the concept. The potassium voltage-gated channels, however, are very slow. So once it hits a particular voltage, they do open or close, but they do it at a very slow pace. So for example here, it slowly closed up, while earlier I think, yeah. So, so we talked about resting membrane potential. Action potential, on the other hand, is this electrical signal that gets carried across that neuron. That's that whole thing we were talking about on the axon itself. The electrical communication occurs across that axon. So it does this by, if there's enough positive ions within the cell, that will end up causing the resting membrane potential to finally hit a membrane potential called the threshold. Okay, so looking at this chart here we have resting membrane potential. So it's negative 70 millivolts. You don't have to remember the numbers again, but I just want you to know it's at this point. If there's more positive ions, right, it's going to become more and more positive or less negative. Does that make sense? More positive ions coming in, right? It's going to become more positive or less negative. That makes sense, right? So at negative 70 millivolts, if there's more ions, notice how it's raising all the way to here at negative 55. So now it's less negative. So there's more. It's like if you're adding numbers to it, right? If you're adding, what is that? If you negative 70 plus what? 15 is it? My math is so bad. 25. Well, whatever the number is, right? It's going to be negative 55 if you add more and more positive charges. Okay, and negative 55 is this threshold. Once it hits this threshold, this is going to cause that action potential. This is going to cause this action potential. If it doesn't hit threshold, if it only gets to negative 60 per se, it's going to just go back to resting membrane potential. Okay, it's not going to cause that spike, that action potential. So it has to hit negative 55 to cause this spike called the action potential. Okay, so threshold, yeah, minimum membrane potential to cause an action potential. Okay, so here's a neuron. You guys remember what are these things called on this side here? Dendrites, very good. Dendrites, what is this thing here? Cell body, very good. And this long extension here? Axon. And then the very ends of this? Yeah, synaptic terminals, axon, no, terminal. Very good question. Usually the axon is longer, the dendrites sometimes are very branched, and this is a bipolar neuron. Don't worry about that. But like a multipolar neuron is easier to identify because they come straight off of the cell body itself, the dendrites per se. And then there's others that are called like pseudo unipolar neurons, meaning that this is the entire thing is an axon itself. There's some dendrites over here, but notice how it's an axon that leads to the cell body, and then once again leaves out of the. It doesn't synapse. Synapses occur from this neuron. If there was another neuron here, okay, here's the synapse over here between the two cells. So okay, very good. You know the different structures. Here is one of those sodium channels or whatever channels, some kind of positive ion channel right here. And here, let's just say it's sodium right now. So if the channels end up opening due to some kind of receptor being activated, then these positive ions will go through the channel into the cell body itself, right? If it's at the dendrites, it might occur at the dendrites, causing them to diffuse all the way over here. But we're just saying it's at the cell body itself now. So as you can see, they are diffusing across the cell body, and they gather. Here, let me go back. You'll see that they diffuse across the cell body, and then they'll gather here at the axon helix. Okay, so you notice how they kind of diffuse out of that location. And once it hits threshold at the axon helix, that's going to cause that action potential, and the action potential will propagate across the entire axon all the way to the axon terminal there. Yeah. Okay. So here, once again, is the resting membrane potential. And once threshold is met, okay, once we hit threshold, that's when we open the sodium voltage gated channels. And when we open the sodium voltage gated channels, where does sodium go again? Out or into the cell? It goes into the cell. If it goes into the cell, what's going to happen to the membrane potential? Is it going to become more positive or less positive? More positive, right? Because sodium is positively charged. What do you call a positively charged ion? Cation, very good. So once these sodium ion voltage gated channels open, sodium influxes in, goes into the cell. So once they go into the cell, that's going to cause it to increase in the membrane potential. So here we see how it hits threshold, causes them to open, increasing the membrane potential. Okay, it does this at a certain point. It's like positive 35 millivolts. You don't have to worry about that. The potassium voltage gated channels actually open as well at threshold. However, what do we say about the potassium voltage gated channels? They're fast or slow? Yeah, they're pretty slow, right? So if they're slower to open, you can't have potassium passing through it because it's not open quite yet, right? It's still closed, right? It's just slowly opening. So once we get to here at positive 35, that's when the sodium inactivation gate, remember that little thing that plugs up the sodium earlier on, sodium gates, it plugs it up, so preventing any more sodium from going into the cell. And that means we're not going to become more positive anymore, right? It's going to stop there. While at the same time, the potassium voltage gated channels now are fully open. And which way will potassium go? They're going to go out, right? Because they're more concentrated inside compared to outside. If they go out, are we going to become more or less positive inside the cell? Less positive, because potassium is a positive ion. So if you have more, if it's going out of the cell, you're losing positive ions. Does that make sense? Yeah. Yeah. Potassium is a positive ion, right? It's more inside. It's going to go out, making it more negative now inside the cell. Okay. So now that they which way would you see this graph go? Now that's over here. If potassium channels are open now, is it going to go up, down, or go straight?
Down.
Going down, right? Because it's going to become less positive. So, yes, goes down. Does that make sense, Elizabeth?
Yeah,
so it's going to be less positive, so it's going to be more negative. So, okay, so once we get here at negative 70, the inactivation gates of the sodium channel now reopen, but it's now in a closed position, so no sodium still goes in or out of the cell. The potassium channels, they now begin to close. Once again, they're pretty slow, right? So they're not going to be closed yet. So there's going to be some still coming through. Not coming. It's still going through it. So some more potassium will be going from inside to out of the cell. So at this point in time, which direction will this? Still going down, correct? Because the potassium channels are not fully closed yet. You're still losing some positive ions. So there it goes. And then there's things called leakage channels. You don't have to worry about it, but that helps reset this back to resting membrane potential. And the leakage potassium channels end up leaking more back into the cell, causing it to go back to resting membrane. But notice how all this process, we're losing a lot of potassium out, right? And we're gaining a lot of sodium inside. But normally speaking, we want to have more on the outside and more on the inside? More what kind of positive? On the inside, right? More potassium inside and more sodium on the outside, right? So how do we reset this? We reset this by having the sodium-potassium pump, right? And what did we talk about the sodium-potassium pump? It pumps what? 3 sodium and 2 potassium. Where? 3 sodium, yes, 3 sodiums out and 2 potassium in, right? Restoring that concentration gradient that we started off with. Does that make sense? Does this use ATP? It's a pump, right? If it's a pump, that means yes, right? Okay, yes, it does. And it's going against this concentration gradient, right? It's going, remember, there's more sodium on the outside, so it's going to go from low to high in this particular case. Yes. Okay. Mm-hmm.
This one says that it goes down, but this one goes down too. This one goes up.
Oh, when I say go down the concentration gradient, I mean from high to low. That's what I mean by going down the concentration gradient. If it's going against the concentration gradient or going up against the gradient, this is, like in this case, where the sodium from low to high in this, yeah. But yeah, normally it's high to low, going down the concentration gradient. No, that's also going from high to low, but it's going from in to out. Yeah. Versus out to in. A lot of different type of concepts, but so looking at this, here we have this. Okay, you tell me, what kind of ions are these? Sodium, very good, because there's a lot more on the outside compared to the inside. And what are these ones here? Potassium, very good. Okay. Oh. You didn't see that. Okay, and do you guys remember which voltage gated channel is this? The one with the inactivation gate here? Is it the sodium voltage gate or a potassium voltage gate? Yeah, the sodium one has the inactivation gate with it. So here's the sodium voltage gated channel, while the blue one is the potassium voltage gated channel. Okay, so now that we established that, ECS means it's extracellular fluid, so it's on the outside, while ICF means intracellular fluid, means it's on the inside of the cell. And here's the plasma membrane here. Okay, so here, once it reaches threshold, right, if there's enough of that positive ions that diffuse across the cell body to the axon helix, and if it hits threshold, what's going to happen again? So what's going to, like, what opens? Which gate? The purple ones is the... Yes, what channels are they? Sodium voltage gated channels open. Very good. Once it reaches threshold, the sodium voltage gated channels open. Very good. So they do open, and once they open, what's going to happen? No, it doesn't close right away. So open. Sodium comes in. Very good. So we have sodium coming in. Okay. At the same time, potassium does open at this time, but they're once again very slow. So they're not totally open at this point. Okay? And once we get up to here, at positive 35 millivolts, what happens there? Not quite. Yeah, it will go down, but we have to first stop the sodium from going in, right? How do you close the door? This inactivation gate, right, will first block the sodium voltage gated channel, right? So that inactivation gate blocks that channel. Now sodium can't pass through anymore, right? Because if it was still open, then more sodium will come down and this thing will keep on raising up and up, right? But we don't want that. So now that it's closed, now sodium can't get through. And notice how the sodium, when it went through, it's going to cause the membrane potential on the inside to become more negative or positive? If sodium came into the... It's going to be more positive, right? If sodium comes in, yeah. If it's positive, what's going to happen to this sodium voltage gated channel then? It's going to thin out. It's going to open. Yes, very good. Yeah, it's going to cause that to open. Does that make sense? Because it's going to reach threshold for that voltage gated channel because there's more positive ions coming in. So that's going to easily cause it to undergo an action potential. So it opens, causing sodium to come in, and so on and so forth. This thing propagates. So on and so forth, right? So this action potential will then propagate across the entire axon now. Okay. It took me forever. No, seriously. It took me a long time. Okay, so now that we're at this point, we said that the inactivation gate blocks the sodium channels now, right? So now, at the same time, we did talk about how the potassium started opening at threshold. But now here, we're finally going to cause it to open, fully open at this point. Okay, so they're going to open, and notice how it's very slowly opening. And now that they're fully open, what's going to happen? What's going to go out? Potassium goes out of the cell. Very good. So potassium goes out, causing the membrane potential to lower, right? So on and so forth. And once we get back to negative 70, it's going to cause it to start to close. Now, the potassium channels are going to start to close, but they're very slow, so they don't fully close up yet, allowing I forgot to mention, but the sodium gates, they reset, right? The inactivation gate opens, but they close up again. And here, this is the slowly closing off part where they don't fully close yet, so potassium still comes out, lowering the membrane potential some more. But finally, they close up. And when they finally close up, then we finally get to the point where some leakage potassium comes in to restore that membrane potential, resting membrane potential, so on and so forth. Okay? Kind of get it? At least has some idea? Yeah? What's going on? Purple door opens, blue door closed, whatever. No! It's not how it is. Okay. So we talked about unmyelinated axons, right? It's slow because you saw that entire process occur, right? And it's always one step leading to another step, leading to another step. So it's actually very slow because it has to open up those voltage-gated channels right next to it and so on and so forth to propagate across the entire axon. Okay? So this is just showing you that over here, this is undergoing action potential here, causing the cells, or not the cells, but the voltage-gated channels right next to it to undergo action potential and so on and so forth. So it's going to have this progress or propagation of this action potential. And unmyelinated axon, once again, we said that it doesn't have those myelin sheath, so I'm censoring that, but anyway. So myelinated axon, in the other case, they do have the myelin sheath, either through the Schwann cells in the peripheral nervous system or the oligodendrocytes in the central nervous system. Don't worry about that. But these here, we have this myelin sheath around it. So what happens is that over here, this is the action potential here, but they skip all these voltage-gated channels over here and go over here to this next node of Ranvier here. So that will cause an action potential there. So basically, they're kind of skipping some voltage-gated channels areas to make this a lot quicker. Okay? So here's my big myelin sheath in the middle. This is kind of show you. So here we have threshold. What's going to happen at threshold again? What's going to happen directly? What's going to happen?
The blue line is going to go up.
Yes, it's going to go up, but how does it go up? It needs to go up. Yeah, how does the sodium come in? The sodium voltage-gated channels, they open. Yes. They open, allowing sodium to come in, right? And then once they come in, it gets to positive 35, like you said. So, and then at positive 35, the inactivation gate closes it up, not allowing any more sodium to come in. And at this point, potassium voltage-gated channels are fully open, right? They were opening before, but now they're fully open. But notice how before when they come in, notice how a lot of them were able to just skip through this myelin sheath and come all the way to this voltage-gated channel over here. Did you see that? And so we're skipping all these potential areas where there could have been voltage-gated channels here to cause the action potential. So we're skipping all of this, going all the way over here to cause that to open. So we're kind of skipping areas of the axon to make this a lot quicker process. Does that make sense? So, yeah, so on and so forth. Causing this to open, sodium rushes in, so on and so forth. Okay. Yeah, and yeah, I guess we could review that. So finally the potassium opens, allowing potassium to go out, right? And once they go out, then it lowers the membrane potential, and then finally leakage channel restores that resting membrane. So now that we have the axon going all across the axon itself, finally we go to the axon terminals. We have the synapse. And the synapse is this location where the axon terminals terminate and then meets up with another cell, whether it's another neuron or the muscle or whatever. But in between, they form a synaptic cleft. And like the name implies, a cleft is a space. So this is a space in between, the axon terminal and its target cell. And this is where that neurotransmitters diffuse across the synaptic cleft. Okay, when I talk about presynaptic neuron, like the name implies, it means that it's a neuron before the synapse. So here's a presynaptic neuron here. And what are these little bubble things again? So here's a synaptic knob here. And there's these little bubbles. No, this is actually within a cell. So these bubbles are yes, very good. The neurotransmitters inside of the bubbles. The bubbles are the vesicles. Remember, they're the membrane-bound vesicles. Okay. So here we have the neurotransmitters inside of the vesicles here. And once they fuse with the plasma membrane, it's going to expel the neurotransmitters as shown over here. So it's going to expel the neurotransmitters into the synaptic cleft. And once they're in the synaptic cleft, they diffuse across the synaptic cleft to that target cell. Okay, so how is this done? Well, the action potential will go all the way to the axon terminal. And once it goes to the axon terminal, there's these different type of voltage gauge channels now. Now it's calcium voltage gated channels, okay? So once it hits the calcium voltage gauge channels, it's going to cause them to open. So we have calcium to come into the cell, right? Because they're more concentrated where? Outside compared to the inside. So that way they go into the cell. Once they go into the cell, it's going to cause a series of events. The vesicles with neurotransmitters are already docked on near the plasma membrane. It's not fused with the plasma membrane yet. But once the calcium comes in, it's going to cause that fusion. Once it causes that fusion, what is that process called again? Where the vesicles fuse and it expels its contents? Exocytosis, very good. And it releases its neurotransmitters, which is the contents of that vesicle. The neurotransmitter would then diffuse across the synaptic cleft to its target's receptor, as shown over here. Here's the target's receptors here. Here's the axon terminal. This is the exocytosis portion of it. The neurotransmitter will diffuse across the synaptic cleft, this little space, and then bind to that receptor there. The receptor might be excitatory or inhibitory, meaning that if it's excitatory, it's going to cause that membrane potential to reach closer and closer to threshold. If it's inhibitory, it's going to prevent it from reaching to threshold. So is it going to be more positive or more negative then? More negative. Very good. Okay, and when we want to stop a neurotransmission from occurring, from stopping that signal from occurring, usually it will be broken down either by enzymes that are within the synaptic cleft, or it will be reuptake by the axon terminal, meaning that whatever neurotransmitter that we found in this synaptic cleft over here, it's going to be broken down. So you break it down with enzymes, either on the postsynaptic cell itself, or it could be reuptake by the presynaptic neuron, so that it could be recycled, basically. So here is the axon terminal here. Here are the vesicles here with the neurotransmitters inside of it. Here's what's the space called? Yes, synaptic cleft. Yeah. Synaptic cleft, and this is just the cell that the neurotransmitter will be sending across. Here's just calcium ions. I colored it purple this time around, and here's the calcium voltage gated channel. Okay, although it's blue, it's not the potassium one. Okay, so it's the calcium one. So we said that the action potential comes right on the axon. It's going to go all the way across to this axon, this synaptic axon terminal. So here we first have the vesicles dock near the plasma membrane. We have the action potential as shown here, coming all the way to where the calcium voltage gated channels are at. And once it reaches the calcium voltage gated channels, what's going to happen? Yes, the calcium voltage gated channels are going to open, and once they open, what's going to happen? Yeah, the calcium will go into the cell because there's more concentration outside than inside. Once calcium goes into the cell, it's going to cause a series of events. You don't have to know this part, but the calcium binds to this thing called synaptotagmin. That's going to end up breaking up the snare protein complexes, allowing exocytosis to occur. So this part you need to know, this exocytosis. Once calcium comes in, exocytosis occurs. And then that's going to cause the neurotransmitter to then do what across the synaptic cleft? To bind to its receptors. It's going to start with a D. Not decompose. Goes from one area with high concentration to low concentration. Diffuse across the synaptic cleft. Yeah, there's a lot of different words. And once it binds, it's going to cause different types of effects, so you don't have to know right now. It all depends on what type of receptors. Alright. There are different types of neurotransmitters, though. One are the amines. The amines are the ones that are derived from amino acids. Okay, there are some amino acid neurotransmitters too. The ones that I want you guys to know: serotonin. Serotonin, it does control our mood, our appetite, and also tends to have, and our behavior as well. It tends to have a lower concentration of serotonin within people with depression. So that's why whenever someone is prescribed medication, it's usually something kind of based on serotonin for people who are depressed.
If you're hungry, do you have high serotonin or was that a controlled appetite?
That's a good question. I got a feeling that lower serotonin levels would probably decrease your appetite.
Decrease your appetite?
Yeah, because you'll... Yeah, I don't know for sure, but that's my impression of it.
That's why some people get fat.
That explains it.
Yeah.
That does explain it. I know people that just go crazy.
Alright. Dopamine, on the other hand, this actually helps us initiate motor control, and it's also our emotional reward center. So, if you think about people with Parkinson's, they tend to have a low amounts of dopamine. So, Parkinson's disease. And so people with Parkinson's, they will probably take something that mimics dopamine, L-Dopa, and stuff like that. And that will help them initiate those motor controls. If you remember Parkinson's disease, they struggle with initiating certain type of motor control. So having increased amounts of dopamine through medication may help them. And the reward center is also really important as well. Epinephrine, also known as adrenaline. Oh, yeah, go ahead, Doug.
I got a shot of dopamine. Is that like
The thing about dopamine, it does multiple effects. We kind of like narrow it down to like the simplified version of it. But was there a reason why you got that shot of dopamine, though? Some sort of test? Huh. Was it radioactive or not? Okay. If it's radioactive, they're just trying to see where it's going to attach. You know, where it's going to activate your brain to see where that activation occurs, and I guess they're going to see whether or not if it's normal or not, type of thing. Is the places that are attaching to it the normal locations as others? I don't know for sure what was the test exactly for, but that's my guess. Like for example, our reward center kind of thing. That would be a normal location where. That is, I think, in our somewhere in our hippocampus somewhere. hippocampus. No, it's not in our hips. It's in our brain. It's a location in our brain, pretty deep inside. It's our emotional center kind of thing, where our emotions are located at. That and the amygdala and stuff. Don't have to worry about that. Okay, that's more anatomy physiology stuff. Okay. Epinephrine is also known as adrenaline. So this stimulates our sympathetic nervous system, which we'll talk about later in this lecture. Norepinephrine, almost the same thing as epinephrine. It's just norepinephrine is actually more from our neurons. Epinephrine is more from our adrenal glands. So norepinephrine also does the same thing, stimulates our sympathetic nervous system.
You said norepinephrine is more adrenal?
No, epinephrine is more adrenal. Norepinephrine is more our neurons.
Okay.
Acetylcholine is a type of neurotransmitter that helps us cause muscle contraction when our nervous system is communicating with our muscles. However, depending on the receptors on whatever cell we're talking about, it could cause excitatory effects or inhibitory effects. Remember, excitatory effects is going to cause the membrane potential to become more positive or negative? Positive, right? Inhibitory, more negative, yeah. There's amino acids. GABA is an inhibitory neurotransmitter. So that's going to cause our membrane potential to become more negative. How would you become more negative? What are some ways? Say again? Work? Oh, yeah, okay. Sorry. I was saying like more on a molecular level. Like what kind of gates would open to cause this to become more negative? So if we open potassium gates, it's going to cause potassium from yes, thank you. Cause potassium from the inside to go out, becoming more negative. Very good. What other ion can we cause to become more negative? Chloride, right? Chloride. If we open chloride, which direction will chloride go? In? Out? In? It's more concentrated where? Outside. Okay, it's more concentrated outside. It's going to come in, making it more negative inside. Very good. So GABA does exactly that. It opens up those chloride channels, allowing chloride to come into the cell, making it more negative. Glutamate is an excitatory neurotransmitter, so that's going to cause our membrane potential to become more positive. Okay, and what ways can we make it more positive? Potassium? Sodium, right? Sodium, because sodium, once you open those, it's going to come in, right? What other ion channels? Calcium, very good. Calcium, and if you think about it, it's 2+ charge, right? So that's going to be even more effective because it's going to double the amount of positive charge coming into the cell. Very good. There are other ways too, if you think about it. For example, we can make, just say glutamate as excitatory, another type of receptor, okay? If we're saying a neurotransmitter that ends up preventing potassium channels from opening. If we prevent that from opening, then it's going to become more what inside the cell? If we prevent potassium from opening, it's going to become more... If we prevent potassium channels from opening, it will become more positive inside, right? Because if it opens, potassium will go out of the cell, making it more negative. Okay? So same thing as an inhibitory effect. If we prevent what from opening, we'll make it more negative then. If it's an inhibitory neurotransmitter, what happens if we prevent what channel from opening to cause it make sodium, very good, or calcium, very good, to makes it more negative inside. So those are different types of way of making it more inhibitor. But to just simplify this opens up calcium channels. And this is opening up, like we said, sodium channels and calcium channels here. Sorry, that's not copper, it's calcium. Chloride channel. Yeah, chloride channel is open for the GABA, allowing chloride to come into the cell, making it more negative. Glutamate causes sodium channels to open, allowing sodium to come in, making it more positive. Or calcium, yeah. Depending on the receptors. There's different types of receptors for glutamate, but both of them are excitatory. Finally, we have something called neuropeptides. And neuropeptides, they are these amino acids, as you can see here, amino acid chains that ends up, for example, like beta endorphins. Those are the type of neuropeptides that help us decrease our perception of pain. So when you have this runner's high, if you guys ever ran, like after you run, you tend to have this euphoria feeling or this pleasing feeling after running. That's that endorphins. The other type of endorphins that comes into play, if you have some kind of traumatic effect, like if you have some kind of injury that causes so much pain and then finally you don't feel that pain anymore, that's the endorphins kicking in to prevent you from feeling that pain. So those are what endorphins are. Okay, so talking about all these neurotransmitters, things that kind of help affect our receptors for those particular neurotransmitters. Caffeine. Caffeine is very similar. It's an inhibitory neurotransmitter. It affects our inhibitory neurotransmitter. It does other things too. So how does it do it? It basically inhibits the inhibitory neurotransmitter. If it inhibits the inhibitory neurotransmitter, that's going to cause our membrane potential to become more positive, right? Double negative is a positive, right? So does that make sense? No? Okay. If it's an inhibitory neurotransmitter, it's going to cause it to become more negative, right? If you inhibit an inhibitory neurotransmitter, that's going to become more positive. And that's what happens in caffeine, right? When we have coffee or whatnot type of thing, it's going to make us more excited. Our hearts increase, heart rate, our breathing rate tends to increase, things like that, because we're inhibiting the inhibitory neurotransmitters. Nicotine acts like acetylcholine. And remember how we said that acetylcholine, it does affect muscles, but in this particular case, it affects different areas of our body that has these acetylcholine receptors. So essentially, it affects the receptors that have excitatory effects. Okay, so for example, it will cause like sodium channels to open to allow sodium to pass through, causing it to become more excitatory. More positive, yes, very good. More pository. Alcohol. It's not really well understood. This is a drug that's not really well understood, but we do know some things about it. It does increase. Huh? Say again?
We should test it.
Test it out. We have to do more testing. Yes, we do to kind of understand this a little bit better. But increases the effects of inhibitory neurotransmitters. So, like GABA. What does GABA do? Remember? It's going to become negative how? So by opening chloride channels. So it's going to enhance that effect. Not only does it do that, it also inhibits excitatory neurotransmitters like glutamate. And what do we say glutamate did? Become more positive by opening sodium and calcium channels, right? But if we inhibit that from occurring, then it's going to become more negative. Yeah, either way, it's going to enhance that negativity of the neurons. And that's why we tend to have slower reaction rates and stuff like that if we're under the influence. Methamphetamines. These end up causing a release of dopamine. It helps release the dopamine. And so remember how we said dopamine is part of our reward center. It affects our reward center. And that's why these drugs are very hard to come off of. So it's very hard. It's very addictive because once you stimulate that reward center, you want to continue to stimulate that. So if you end up, people have bad withdrawal symptoms because they're addicted to it. Yeah. Mm-hmm. Uh-huh. Yes, you naturally release less dopamine. If you think about it, that makes sense, right? Because if you're taking these drugs and those are mimicking dopamine, your body is not going to, it's going to say, oh, we have enough. I don't have to work anymore. I don't want to produce any more dopamine. So it stops releasing dopamine, period. And so, and then not only that, you end up having these receptors that accept dopamine. You're going to end up upregulating because there's so much dopamine out there. It's going to say, okay, we need more receptors to, remember those protein receptors and those ligands, how we increase the receptors to increase the saturation. So if we increase the receptors, we're going to become more and more, adapted to that certain amount of dopamine. And so you want to increase the dosage to get the same high effect, kind of thing. So that also comes into play.
So it doesn't release, your body doesn't get used to the same amount of dopamine, but it just releases less?
It does both. It releases less dopamine, and plus, since you upregulate the amount of receptors, it needs more dopamine to be able to have the same effects. Yeah. Say again?
Are those your fingers in the methamphetamine?
My fingers? I don't know. No, that's not my fingers. Sorry. Cocaine. So these inhibit the dopamine transporters from reuptaking dopamine. So if you prevent the reuptaking of dopamine, then is there going to be more or less dopamine in the synaptic cleft? More, right? Because you're not reuptaking anymore, so it's going to stick around, right? So that's going to, once again, cause our reward center, make it a very addictive type of drug. Yes. LSD, it acts on serotonin and also dopamine receptors. So similar type of effects. It does cause hallucinogenic kind of effects as well. Marijuana. Okay, these inhibit the inhibitory neurotransmitter for dopamine. So what's going to end up happening? If you inhibit the inhibitory neurotransmitter for dopamine, then it's going to cause dopamine to be increased in releasing it. Correct. So increase the amount of dopamine in the synaptic cleft. So there is some addictive properties to this as well. Opiates like heroin, these end up binding to the endorphin receptors. And what do we talk about endorphins? What does endorphins do?
Make you happy. Or
Yeah, it does kind of, but yeah, it decreases the amount of pain, right? So if you think about it, if you have heroin or opiates, that's going to do the exact same thing. It's going to end up binding to these receptors, so you don't have to feel the pain. So it also does this inhibitory effect on the inhibitory neurotransmitters. So it's going to stimulate the amount of dopamine involved. So that's why these are also very addictive as well. So yeah. And something like so illicit drugs like heroin is an opiate. If you say in the medical field, you use things like Valium or morphine and stuff like that as a pain suppressor. Okay, let's pause for a little bit since I know we haven't had a break yet. Alright, so for the central nervous system, what does it consist of again?
Brain and the spinal cord.
Yes, brain and spinal cord. Very good. So the brain is mostly contains a lot of different centers: homeostatic, sensory, motor, emotional, and intellectual centers as well. The spinal cord communicates information from the brain to the rest of the body, and from the body to the rest of the from the rest of the body to the brain. So it communicates back and forth. So one of the information is sensory information. So if it's sensory information, we're having information from our what to our what? Body to our brain. Very good. Motor, on the other case, when we're actually going to cause some sort of action, we're brain to the body. Very good. Okay, on our central nervous system, it's surrounded by a couple of things. Meninges, these are just connective tissue that help protect our brain and spinal cord, although this picture is blocking it. There we go. And you can see it on this particular diagram here. All this is the meninges. It has different layers to it. Just know that it helps protect the brain and surrounds the brain and spinal cord and helps protect them. Also, there's cerebrospinal fluid, CSF. This is the fluid that surrounds both the brain and spinal cord to help cushion it. So if we have some sort of impact, our brain doesn't hit against the skull. It helps kind of buffer it a little bit. Every now and then, if you have a big enough impact, it's going to hit and cause a concussion. It also supplies nutrients, hormones, different types of white blood cells and stuff like that to the brain and spinal cord. So when people talk about meningitis, we're talking about inflammation of the meninges. Yes, meningitis, right? So it's inflammation of the meninges, and this is those connective tissue that surrounds the brain and the spinal cord. Okay. Although, one of the causes of meningitis is due to bacteria that is spread through the brain through the cerebrospinal fluid. So, yeah. It could cause, definitely irritation. Like, you'll have definitely high fevers and stuff like that because it's a bacterial or viral infection and stuff like that. And it causes swelling as well. And so if you have swelling and putting a lot of pressure onto your brain, that's not a good thing. It could cause brain damage if it gets to that level type of thing. It most likely would be able. It has the capabilities to spread. I would believe so. Yes.
Does that cause hemorrhaging?
Hemorrhaging, I think, is aneurysm. I think is different. That's called a blood vessel bursting kind of thing. Okay, the brain stem of the brain consists of the pons. The pons is located over here. The medulla oblongata, or just the medulla, is just this portion of it. And what did they say in Waterboy? I don't remember.
When they ask why the crocodile, why they like ate cookies in that part, I remember that.
So the pons, medulla oblongata, and the midbrain here. So this is all a part of the brainstem. Its function is to help allow some information to pass through the brain. It selects what kind of information to pass through the brain. It helps us regulate in our sleep and arousal, and also help with coordinated movements as well. The cerebellum, located over here, receives sensory information from our limbs and also the muscle length as well, also with auditory and visual information. This is all to help us coordinate our balance and movement. Remember how we were trying to balance on one leg and we try to balance it without with closing our eyes and stuff like that? All that information goes into our cerebellum, either visually or our lengths of our limbs and our location of our limbs and the lengths of our muscles and stuff to help us balance. Is used by the cerebellum. The thalamus, located over here, helps us relay all the information, either from the brain or senses to our brain, or from our brain to the different parts of our body. So it's a relay center, meaning that all this information has to cross to the thalamus, and the thalamus will determine where it will end up going. So, for example, if we see something, so we have sight information going to the thalamus first, and then from the thalamus it will go to sending to our occipital lobe, which is back here. So that's as an example. Or if we're talking about from the brain, we have part of our brain will send information to the thalamus, and then that thalamus will send it to our limbs to move as for motor control. The hypothalamus is right underneath the thalamus here. Is this thing that I'm kind of coloring now. The hypothalamus does multiple things. It helps regulate our temperature, blood pressure, hunger, thirst, sex drive, sympathetic response, emotional pleasure center, and also helps control our pituitary gland. And it also is involved with our biological clock. So it does a series of things. When we're talking about how we regulate body temperature, whether through sweating or shivering, it's controlled by the hypothalamus. The cerebrum is basically everything else here. All of this is this part of the cerebrum. And it has multiple functions as well. Helps us to reason, helps us with our language, personality. It also helps us to perceive sensation as well, and also help us to regulate our body movements. Normally speaking, generally speaking, our right hemisphere, so over here, our right hemisphere here, controls the left side of our body. Because due to some decussation, meaning that some of the neurons will end up crossing over to the other side of our body. So information received from that side of the body will go to the right hemisphere as well, while the left hemisphere will end up controlling our right side of our body, and so on and so forth. Our right hemisphere tends to be stronger in spatial recognition. So if you play video games or whatnot type of thing, it helps you do that. Also for pattern face recognition as well, and also to be able to read faces as well.
So are left-handers smarter?
Not necessarily. I'm just saying like that they tend to have stronger at doing these particular things.
Doing what thing?
Like spatial recognition, pattern facial recognition, and being able to read emotions. Doug? Say again? It could be. The thing about this is generally speaking too, meaning that not everyone's right hemisphere will have these particular characteristics. Some people, like about 10% of the people, have this actually on the left hemisphere kind of thing. So it really depends on the person. The left hemisphere tends to deal with language. We have the Broca's and Wernicke's area where it helps us be able to understand language and to speak. And it tends to have stronger parts of logic and also for mathematics as well. All right, the peripheral nervous system is divided up even more so. We have the sensory division, and this carries sensory signals from our body to the central nervous system. The motor division, like we've been talking about, carries information from our CNS to our body. Yeah, muscles, very good. Muscles and glands. So when we're talking about the motor division, there's different parts of the motor division. There's the somatic motor division, where we're actually voluntarily controlling our skeletal muscles. So like, I forget this guy's name. I've never seen the movie before, so just know about it. But anyway, so like Napoleon Dynamite here, he is voluntarily controlling his muscles to do this dance move. So this is part of the somatic motor division. It's also involved with our involuntary reflexes regarding to our skeletal muscle. Guys, name a reflex that we did? Yeah, the knee-jerk reflex or the patellar reflex. Okay. Autonomic nervous system, like the name implies, this is due to our involuntary or automatic muscles, so smooth muscles and cardiac muscles. So those, when we're talking about smooth muscles in the digestive system, we have this process where we're moving food down the tube called the, it goes down the esophagus through what process? It starts with a P. Peristalsis, very good. So that's utilizing the smooth muscles to cause that to occur. Okay, cardiac muscles for our hearts to pump. So involuntary control, yeah. So when we're talking about autonomic nervous system, we have the sympathetic nervous system. This is the fight or flight, right? Meaning that what's going to happen, like, to this little girl? What's going to happen to her heart rate? It's going to increase, right? Respiratory rate. Increase, right? Digestive. Her digestive system. What it's going is going to increase the amount of digestion occurring or decrease? Does she really want to be digesting right now? Does she really? Yeah, it's going to decrease the digestion, right, from occurring. Very good. So how about, I was going to say, how about your pupil? Dilate, right? If you ever scared a cat or something like that, you'll see that their pupils dilate. Or even if you scare a person, you'll be able to see that. Parasympathetic division, on the other hand, does the exact opposite. So it tends to be this rest and digest part of the autonomic nervous system. So in this particular case, the polar bear's heart rate will be decrease. Their blood pressure will be low as well. How about their digestion? Yeah, increased activity of digestion. Very good. So that's how it divides, right? We have the central nervous system consists of the brain and spinal cord. We have the peripheral nervous system, and we said that it has one part is for the sensory, right? And the sensory portion of it, we didn't really discuss too much about it, so you don't have to know. But the motor division is divided into so many different ones. The somatic motor division controls what? It's what this Napoleon Dynamite guy is doing. Voluntary muscle control, very good, including our reflexes as well. While our visceral motor division controls our autonomic nervous system. So we're talking about sympathetic division, which does what? Fight or flight, while parasympathetic does rest and digest. For our autonomic, so we're talking about cardiac, smooth muscles, etc. Okay, now going on to senses now. So how do we get sensory input? We get it from receptors that help detect our environment, sending signals to our CNS. So there are different types of receptors. There are thermal receptors. Thermal receptors, like the name implies, detect what? Yeah, heat or coldness. So it depends on the temperature, and that's what it's going to respond to. Photoreceptors. Photo is for light. Nociceptors. These are the ones that receives information of pain. Okay. Chemoreceptors, depending on the chemicals. So you probably don't know, but odor and taste are actual chemicals that cause these receptors to be activated for us to smell or taste something. And also depending on our body fluid composition as well. So when we're detecting something like pH is also a chemoreceptor that will detect that. Mechanoreceptors help us respond to physical deformation. So when we're actually putting pressure, vibration, touch, these kinds of different things. The ones that are less known or not as obvious are hearing. That's actually a mechanical receptor, and also equilibrium. When I talk about equilibrium, it's like being able to know about our orientation kind of thing. So when we're actually like doing flips, or if we're accelerating, decelerating, that's part of equilibrium. Are you guys? Yeah, he's mechano.
Thermoreceptors heat, photoreceptors response to light, nociceptors response to pain, chemoreceptors.
So when we're talking about the eye, we already talked about it a little bit before, but this white of our eye is called a sclera. It's just this outer surface of our eye. The cornea is this anterior or front part of our eye that's actually clear, right? Allowing light to pass through. It also helps us to focus on certain image too. The iris, as shown over here, gives us the color of our eyes and also helps to control our diameter of the pupil. So if it dilates, it allows more light to pass through. If it constricts, it ends up reducing the amount of light to pass through. Oops, sorry, I went backwards. The pupil is just the space here. That's the location where light will be able to pass through, so that we could end up having an image formed on our retina on the back. The lens over here help us focus whatever image or light rays that pass through. The retina is this yellow layer here. And remember, the retina contains cells that actually detect light. Okay, it actually has those photoreceptors. And when we're talking about photoreceptors, what are the two types of photoreceptors, you guys? Cones and rods. Very good. Over here at the this information from the cones and rods will be sent to the optic nerve here. That will give us information that will be sent to our brain to show us or perceive vision. Over here, where the optic nerve and the retina kind of coincides, at that location there is no photoreceptors, forming that blind spot. And hopefully most of you kind of noticed where your blind spots were at, for you guys who did it 2 weeks ago. Yeah? You don't have any? Okay, so different types of photoreceptors. Rods, once again, they're very light sensitive. However, they're not very good in perceiving colors, though. And so you can't really make out colors for these. We have a new friend here, Samira. Say hi to my class. You can sit down. Cones, on the other hand, are very easy to distinguish color, but they don't function well with low amounts of light. So our rods help us have that night vision, while our cones just help us with color during more amounts of light. There are different types of cones: red cones, blue cones, and green cones. And of course, each one responds to a specific type of wavelength to help us see those particular colors. People who are have color blindness, they lack a certain type of cones. So the most common one are the red-green color blindness. So for people with red-green color blindness, they wouldn't be able to see this number here, which 16. Yeah, some people. I mean, it's not uncommon to have people who are colorblind. The interesting thing about the rods and cones, as you can see, as light passes through the eye, they pass through these cells here, and here are where the rods and cones are located at. So it's actually passing through some translucent cells to be able to hit our rods and cones, and then that's going to send information to our optic nerve. You guys can tell what kind of photoreceptor is this? Rod, very good because of the shape, right? Versus this over here is a cone. Yeah, very good. Okay, visual acuity. People with normal vision have 20/20 vision. People with 20/10 vision, these are people who can be able to see things at 20 feet away, while normal people see it at 10 feet. So people who have normal vision need to be up closer to see the same thing as someone who has this 20/10 vision. So, yes. Yes, 20/10 is better than 20/20. Very good. No, you're right. You're right. So that, which means that what you can see at 15 feet, what you can see at 20 feet, other people, normal people, can see at 15 feet, right? They have to get closer to be able to see it. Yeah. People with 20/50, on the other hand, that means that you can see at 20 feet you can see at okay, so yeah, what normal people can see at 50 feet, you can see at 20 feet. Does that make sense? So normal people can see it a lot further, while you need to be closer up to 20 feet to see it. Nearsightedness is like people with that 20/50. It's also called myopia. It's due to having a longer than normal eye. So notice how a normal eye will be over here, and notice how this is actually an elongated eye. And so the point of focus is actually a lot nearer and doesn't hit the retina. And that's why we need some kind of corrective lens to help bend the light to be able to hit it, that point of focus at our retina, to be able to focus. You cannot see far away, so you can only see near things. Okay. So people with farsightedness or hyperopia, their eyes tend to be shorter than normal, so their point of focus is actually beyond the eye itself. So they'll have some sort of corrective lens to help focus it, so that it will hit the retina to be able to stay in focus. Okay. Astigmatism, these are people with a warped kind of eye where so it's misshapen, so that you will have blurred vision. So corrective lenses do help bend that light to help correct that. Oh, I mean, usually, no, it's like within our DNA. So as we're growing, it could form, kind of thing. Because sometimes we're not born, like when I was small, I didn't need glasses, But as I was growing, my eyes tend to elongate, and then that's why I need to wear glasses to be able to see things far away. Okay, so structures of the ear, we have different structures. We have the pinna, which is just the flappy portion of our ear. I should have put a Dumble picture, I forgot. But anyways, we have the auditory canal. It's just the passageway to hit our eardrum. The eardrum is the thing that actually vibrates when sound waves gets to it. And so that's all part of our outer ear. Within our middle ear, however, we have these bones. And these bones are very tiny bones. I wish I could show it to you. I do have some in the anatomy class. Maybe sometime when we have time, I'll show it to you. But they're very small. They have the malleus over here, the incus over here, and also the stapes. Yeah, inside our ear. Deep inside. So these actually vibrate as well. So when the eardrum vibrates, it's going to vibrate these bones. Then that's going to cause us to have that vibrational sound. And once again, what kind of receptor is this then? If it's detecting vibration, Optimus Prime, Mechanoreceptors, very good. Yes. Transfer. So there's this tube here that connects our middle ear to our pharynx, and it's called the eustachian tube. We have it there so to help equilibrate the pressure. So when we're going up high places, you notice there's a buildup in pressure in your ears, so we tend to open our mouth really big to equalize that pressure. When you open your mouth, that expands the eustachian tube to be able to equalize that pressure from the middle ear to the pharynx. The inner ear contains the cochlea. The cochlea is for hearing, and within the cochlea itself, there's this receptor here called the organ of Corti, and that's our hearing organ. They have these little hairs here that vibrate when sound waves pass by it. Yeah, silly. The semicircular canals, as shown over here. So there's one semicircular canal here, another here, and a third one here. They detect our balance and equilibrium. And so this is for angular acceleration. So someone who is doing this, what do you call it, cartwheel, thank you, will help stimulate like this anterior semicircular canal. While someone who's spinning like this will be stimulate another semicircular canal. And finally we have the horizontal one if you were to flip upside down and twist like that, but okay. The vestibule is this portion. Oh, where have the vestibule is this portion of the organ here, and this actually helps with our acceleration detection. So when you accelerate or brake, this is what detects it. And also, if you have so that's the acceleration portion, linear acceleration. And also up and down movement. So if you're going up and down the elevator, like little plucky duck here, some of you are like, who in the world is that? Yeah. But it was a funny skit back in the day. So we're going from the nervous system. It helps control our muscular system, right? So our muscular system, we're talking about skeletal muscles, right? Skeletal muscles are voluntary or involuntary? Voluntary, yes. So, and the name implies that it's attached to our skeleton. It's attached to the skeleton due to these tendons here that help attach our muscle to the bones itself. So these are the tendons, this white portions of where the muscles attaches to the skeleton. A muscle fiber is the actual cell, muscle cell. It has many nuclei, as you can see over here. And it's due to when we're developing, we have multiple cells fuse into one cell. The sarcomere is the functional unit or contractile unit here. It's made up of thin filaments, as shown over here. These are the thin filaments. The thin filaments are mostly actin. Okay, so these are the specific type of proteins that make up the thin filament. And then we have thick filaments called myosin, and these are mostly myosin. That is, these are the thick filaments, and it helps with the contraction. We'll go into that. So when we contract, our muscle sarcomeres shorten, okay? We're not actually shortening the thick and thin filaments. We're actually just shortening the sarcomeres. They're going to just overlap with each other. So this part you don't have to know, but I just want to kind of throw it out there so you guys have a reference. So there's an action potential that occurs across the neuron. It then causes neurotransmitter. Do you guys remember which neurotransmitter affects skeletal muscle? Starts with the A. Acetylcholine. Yeah, ACH. Acetylcholine is released. That's the neurotransmitter. It's going to bind to acetylcholine receptors on the muscle itself. that's going to eventually cause calcium channels to open, allowing calcium to come in. And so this calcium will bind to this protein called troponin. And what troponin does, it actually causes the tropomyosin to unbind from the actin, so it reveals the actin sites. Okay, and then when the actin sites are revealed, then myosin can actually bind to it. And myosin at this time will have ADP and what is PI again? Yeah, what kind of phosphate? It has no carbon or hydrogen, so it's inorganic phosphate. Yes. Okay, so inorganic phosphate and ADP at this time are bound to the myosin head. So that's going to cause that attachment to the actin. Okay, so over here you have the ADP over here, and the inorganic phosphate are actually bound to the myosin head, allowing them to be able to bind to the actin. The actin is this thin filament here, this blue filament. It's going to form this cross bridge. When the inorganic is released, phosphate is released, thank you, then it's going to cause this power stroke. So this is going to end up causing this to contract, well, not contract, but it's going to move, right? That's going to pull the sarcomere to become thinner, so it's going to be shorter. So it's going to cause it to slide. The ADP dissociates from the myosin head, and then when that happens, ATP will now bind to it, bind to the myosin head. Once it's bound, the thing is the myosin head has an ATP ACE. So it has an enzyme that breaks down ATP into ADP and inorganic phosphate. So when it binds to ATP, and then at that point in time, it doesn't actually bind to actin. It's not until we break it down to ADP and inorganic phosphate that we repeat this process where it actually binds to actin to potentially cause another contraction. So in an animation's point of view, this is what it looks like. Here, this purple line is the thin filament. So it's made up of what type of protein again? It starts with the A. Actin. While this thick filament here is made up of myosin, and this is the myosin head here. And notice at this point in time, an inorganic phosphate and ADP are bound to it. This is calcium, and like I said, calcium binds to troponin. This part you don't need to know. It's going to cause the tropomyosin to move away, allowing access for the myosin head to bind with the actin. So here it's bound now. Okay. Once it's bound, the inorganic phosphate leaves, and when it leaves, it's going to cause this power stroke. It's going to cause the movement of the actin. And ADP then dissociates, allowing ATP to come in. And once ATP comes in, it's going to unbind from the actin. Okay, remember how I said the myosin head has ATP ACE? That's going to break down ATP into ADP and inorganic phosphate again, allowing us to rebind to the actin again, right? And the process repeats. And when I said that power stroke occurs, the power stroke basically does not make the thin and thick filament any shorter, but it does make the sarcomere shorter. The sarcomere is from here to here is the sarcomere. So it's going to just pull these Z discs, these blue lines, closer together. So it's going to be like that. And that's what causes our contraction of our muscle. Okay? So just to kind of re-emphasize this portion of it. So if someone's dead, right? Like zombies. Okay? We're saying zombies, right? What happens is that does dead people have ATP? No, right? So we have this portion where normally when ADP unbinds, ATP binds to it, the myosin head, causing the unbinding of the myosin head to the actin, right? So if we don't have ATP, that means this is going to happen, right? So it's going to be bound. The myosin head is going to be bound to actin, and that's called rigor mortis, right? To occur. But rigor mortis, when the people who are dead, they tend to stiffen up due to this process because there's no ATP to unbind the myosin head from the actin anymore. The muscle is not, it's not really contracted, but they're just frozen in place, kind of thing. It's going to be harder to pry off if they're holding something. However, given enough time, these actin and myosin proteins, they're going to degrade. Okay, so when they degrade, then the muscle degrades itself, so they're going to loosen up again. Okay, so it's only a period of time. Go ahead, Elizabeth. I think she's on. Oh, that's a reflex, actually. If they jerk all of a sudden, they might be brain dead, but their reflexes are still in place. So that must be a very fresh person still who just, So the reflexes, remember, they don't need the brain for it to control, right? It goes to the spinal cord and straight back. So even though they might be brain dead, the reflexes are still occurring. So if you put a hot plate, I don't know why you would do that to a person who's brain dead, they will still reflexively contract and move away from the hot plate. Okay. And I think that's, okay, almost done. Okay, somatic motor neurons. So from the spinal cord, the somatic motor neurons will send information from our brain to our muscles, and so for us to contract. So these somatic motor neurons can innervate not just one muscle fiber, but could innervate multiple muscle fibers too. So motor neuron is when we have one motor neuron, as shown over here, like the blue one, innervating all the muscle fibers that it innervates. So the blue ones, all the things that are in blue is one motor unit, while all the things in red is another motor unit. So pretend this, okay. So it could innervate multiple muscle fibers, multiple muscle cells. Or it can innervate very few of them. It just depends on how fine focus of a muscle we want. Or so people with muscles with fine control, we tend to have lesser amount of innervation. So we have motor neurons that innervate fewer amounts of muscle cells. Does that make sense? So it has better control, more precise control. If you think about it, it's like if you have these motor neurons here that will innervate just a single neuron, innervating a single muscle fiber. That'll help us have more precise control. Kind of like our eye movements. So our eye movements tend to have very fine motor control. So there's more neurons per muscle fibers. Does that make sense? It's more like a 1-to-1 ratio. Versus something that has more strength involved, that means one neuron is innervating many muscle fibers. And that way you have more, it tends to have less control, but it has more strength in it. Because when you tell that one motor neuron to contract, it's going to contract many muscle fibers at once. And that's going to increase the amount of strength, but less control. Does that make sense? The acknowledgements and pictures. Okay.